Plant breeding. Green Revolution was dependent to a large extent on plant breeding techniques for development of high-yielding and disease-resistant varieties in wheat, rice, maize, etc. Plant breeding is the purposeful manipulation of plant species in order to create desired plant types that are better suited for cultivation, gives better yields, and are disease resistant. The main steps in breeding a new genetic variety of a crop are: A. Collection of variability. Two most important steps in plant breeding are to create genetic variation and selection. Variation means differences among individuals of a population or species for a specific character. Genetic variation. It is the source of raw material for selection. These are heritable due to genotype and are transmitted from one generation to other. Such variations are useful in selection. B. Evaluation and selection of parents. A newly developed variety is evaluated for its yield, quality, disease, and insect resistance before it is released to farmers. Indian Council of Agricultural Research (ICAR) carries out the evaluations in India. In every agroclimatic zone, every crop is evaluated for minimum 3 years. A new pure line, population or hybrid that is superior to the existing varieties as well as to other new material may be released as a new variety. C. Cross-hybridization among the selected parents. The most frequently employed plant breeding technique is hybridization. The aim of hybridization is to bring together desired traits found in different plant lines into one plant line via cross-pollination. The first step is to generate homozygous inbred lines. This is normally done by using self-pollinating plants where pollen from male flowers pollinates female flowers from the same plants. D. Selection and testing of superior recombinants. The selection process is crucial to the success of the breeding objective and requires careful scientific evaluation of the progeny. These are self-pollinated for several generations till they reach a state of uniformity so that the characters will not segregate in the progeny. E. Testing, release and commercialization of new cultivars. This evaluation is done by growing these plants in the research fields and recording their performance under ideal fertilizer application, irrigation, and other crop management practices. It will be followed by testing the materials in farmers' fields for at least 3 growing at several locations in the country. Wheat and rice. In 1963, several varieties such as Sonalika and Kalyan Sona, which were high-yielding and disease-resistant, were introduced all over the wheat-growing belt of India. Semi-dwarf rice varieties were derived from IR8 and Taichung Native 1 were introduced in 1966. Later, better-yielding semi-dwarf varieties, Jaya and Ratna, were developed in India. Sugarcane. Saccharum barbari and Saccharum officinarum were crossed to get the desirable quantities of high yield, thick stems, high sugar and ability to grow in the sugarcane areas of North India. Millets. Hybrid maize, jowar and bajra have been developed in India, which are high yielding and resistant to water stress.